I will talk about how we use brain network modeling to really understand how human cognition, the brain function, not meaning activity, but really function in terms of intelligence works. And this is very new stuff from our lab. I'm really happy to share that with you. Before I show you that, I want to show you a little bit of previous work, but only very shortly. So you all know it is about brain network modeling, personalized brain network modeling, and eBrains enables and brings the technology to facilitate integration of different data types to build these personalized brain network models, to bring them to the patient at the end. And Paul made that very clear. We have a proof that our research makes sense if we can help with our research results, with our innovations, the patients. This is the best proof that we did something useful, meaningful, that is right at the end, and not just another paper. So this was, by the way, a colleague of mine, Jessica Palmer, and I use this just to show this personalization aspect. What you see here is also the brain of Jessica, but this comes from a study that has been published a couple of years ago in eLife, where we simulate, I think, 14 individual virtual brains of 14 adults. And we optimized or fitted the model, the brain network model, by fitting the region-wise fMRI time series. So we had the fMRI activity of these individuals. We also had EEG of these individuals. We had simultaneous EEG fMRI. And we optimized the parameters by fitting really the time courses, not as usually done, the functional connectivity matrix or average functional connectivity matrix, or now functional connectivity dynamics matrix, but we really use the time series. And what we gained was a model that was able to reproduce for 14 individuals the resting state activity, fMRI activity, over 22 minutes. And for each individual brain region of these models, we had the neuronal population model consisting of excitatory and inhibitory population with all the related state variables of the model, the firing rates, the local field potentials, the inhibitory excitatory input currents. So by reproducing the 22 minutes of fMRI resting state activity for each individual, we could infer the underlying detailed neuronal interactions and features. So for example, here you see from the occipital region the ongoing excitation and inhibition firing. And thus we have for each of the regions, and of course we couldn't measure it non-invasively and even not invasively because we could not reach all the brain regions at the same time with electrodes. So we use the brain models to construct a model that generates the observables, the measurements, and at the same time gives us a candidate theory what the underlying neuronal interactions are. And we have a lot of state variables that we could look at, and I only show here a couple of more state variables. So here you see the resting state fMRI that can be simulated. Here you see the input current, synaptic activity. Here you see the local field potentials. Here you see actually we inject artificial activity to then use these optimized individual models to understand if we inject, for example, alpha activity, how would the system react, how would the firing rate in the different brain regions react, and how that transfers then, for example, to the fMRI signal. We see here in response to this amplitude modulation of the alpha rhythm, the modulation of the excitatory and inhibitory firing rates, and this translates then into a slow modulation of the fMRI signal. So now in the next slide, this is very complex and probably you cannot see it, but it's also in the paper. So in the upper panel you see all the simulated signals, in the lower panel you see the corresponding experimental data, and they are sorted according to this time scale. So here in this panel you see the relation of firing rates to the phase of a single alpha oscillation, which lasts 100 milliseconds. And here on this end, you see the slow switching of the spatio-temporal activity patterns over 22 minutes, so the switching of functional connectivity matrices. And in between, you see, for example, the behavior of inhibitory and excitatory postsynaptic currents in the simulation and in animal recordings. You see the relation between the firing rates and the power of the algorithm, EG and fMRI and so forth. And we can compare this from our model. So these are all different analyses of the state variables of our model, and we compare these results with the actual experimental data. For the experimental data, we have only snippets of observations. Yeah? They are all disconnected, but with the model we can now put them together in a single model, and now they are causally and mechanistically linked. So if we would manipulate or change, for example, the ratio of amplitudes of inhibitory and excitatory postsynaptic current, then this would have an effect on all the other variables as well. And this gives us a causal model and not just a correlative model as we could build from the empirical data alone. And so the model allows us to really understand the principles, the mechanisms of the interactions across the different spatial and temporal scales. Now, this is all about the activity, but we want to understand at the end how human cognition works. And this is a challenge, of course. So the first question is how do we define different concepts of human cognition? So what makes up, for example, human intelligence? And if we think about this, we will see that in order to act smart in our world, we have to solve two categories of different problems. The easy problems that we can solve very quickly, and then there are the more complex and challenging problems for which we need more time to solve them. And this is one example. So if we see that a traffic light turns red, then immediately we take the decisions. We don't have to think long about it and step on the brake, for example, or stop walking and crossing the street. But if the challenge is to find the best route in this metro map, the shortest or fastest, whatever, then we have to look into this, we have to analyze the map and come up with a solution. And this will take a couple of minutes. Or if we write a big proposal, if we write the eBrain Surf proposal, for example, then this is a big, hard problem that needs a lot of reasoning and cannot be solved quickly. So these are the two categories. So how would you define now intelligence, the ability to act smart in our world? So of course there are others who have thought about it, but would you have a quick answer what intelligence, human intelligence is? What is the most important aspect, or are the most important aspects of human intelligence? If you don't want to say something, I have a definition for you here, so no worries. And it was not easy because there's a lot of debate, but I read it for you, if I can. Actually, the light is very bright, so you cannot read it for sure. What is intelligence? A broader definition was agreed by 52 prominent researchers on intelligence. Intelligence is a very general capability that, among other things, involves the ability to reason, plan, solve problems, think abstractly, comprehend complex ideas, learn quickly, and learn from experience. It is not merely book learning, a narrow academic skill, or test-taking smarts. Rather, it reflects a broader and deeper capability for comprehending our surroundings, catching on, making sense of things, or figuring out what to do. Intelligence so defined can be measured, and intelligence tests measure it well. So we have to believe that, but for sure there are many tests available, and it has been found that interestingly there are of course many different domains that can be tested that contribute to the ability to act smart in our world, to intelligence. And here are a couple of keywords for those tests. So each line represents one of those cognitive tests, and there are many more. These are just a few examples. What has been shown is that across different domains, so for example, memory or the knowledge of vocabulary, or spatial orientation, many other things that can be tested. Sensory perception is also part of intelligence testing. And usually there is a good correlation. So individuals who perform well in one cognitive domain tend to perform well in all others. And you see this here, that there are always positive correlations between these tests. So this is interesting and useful, because this means that we could actually have an operational measure of general intelligence. And this is exactly what people thought. So they came up with a general factor of intelligence, the so-called G factor. And the concept is to capture the broad ability to act smart in the world. And this means to capture the performance in these different cognitive domains and others. It's just a selection, which are measured with many different tests. And these are also just only examples, but it is to explain the concept of general factor of intelligence. So the general factor of intelligence, G, is correlated to the different cognitive domains and to the different tests that test the ability or performance in the different cognitive domains. Interestingly, as you can see here on this panel, G, the general factor of intelligence, is anti-correlated with age. Yeah? So when you are aging, then many of these cognitive abilities are declining. For example, the ability for memory, or also the processing speed, which is also a feature, that they are declining. There are others which actually increase. So for example, the knowledge of vocabulary is still growing over the age. So there's a positive correlation, as you can see here. But in general, age and the general factor of intelligence are anti-correlated. So there's a decrease of G over aging. So then, two other important concepts, the concept of fluid versus crystallized intelligence. I think everyone is aware of this. Fluid intelligence means the ability to reason, to solve problems, independent of declarative knowledge. And crystallized intelligence is basically the knowledge that we can acquire. And this is very much, of course, dependent on the cultural background and so forth. So now, in order to test the ability for reasoning, for deep thinking and problem solving, the people came up with a couple of tests, and this is one which is used very typically, the Ravens or Penn progressive matrices or PMAT test. And what does it involve? So what you want to avoid in these tests is that you need declarative knowledge, because then it would be dependent on your background, your culture, education, and so forth. So for this pattern completion test, this is not necessary. And the tests are built such that they are having an increasing difficulty. So you see here two tasks. One is probably the first one, a very easy one. And the task is to complete the last figure and find the hidden rule in these patterns. I'm pretty sure some of you found it already, so for me it was also easy to find. But here is another example, and this is a more difficult one. And to be honest, I haven't solved it so far, but I also haven't spent a lot of time on it. So if one of you knows the answer, please tell me, then I know it from now on, but I couldn't figure it out. So anyways, it doesn't matter, it just shows you the concept of how this is tested. And what does it mean? What exactly is happening in our brain if we have to solve such complex problems by reasoning and deep thinking? We have to split the big problem in many small problems. Yeah? And then we solve each small problem, and then we have to keep the results of the small problems in our working memory and need to integrate them in order to solve the bigger problem. So we need not only to take decisions and come up with answers, but we also need to keep stuff in our working memory. and to integrate it, and maybe then notice it doesn't fit and come back, start analyzing the sub-problems and so forth. And this can be a very deep hierarchy in some cases. And in contrast, if there are simple problems, then we don't have to do this integration. Yeah? We have a sensory input and can directly react. And there, actually, it would be harmful if we would start to integrate a lot of additional evidence, so start looking at the context. So in which setting do we have the traffic light, and what year do we have, and so forth. Then it would take too much time. So in this case, we really want to be flexible and respond active promptly. And this concept of taking fast and slow decisions has been researched before, and one prominent researcher in this field is Daniel Kahneman. This is this person. I think he was born in the 30s, and he won a Nobel Prize on his research on decision making. I think in 2002. So he also wrote that interesting book. The title is Thinking Fast and Slow. And he came up with a theory that we have two systems. System 1 operates automatically, and it's for the easy decisions and easy problems, simple problems. And System 2 allocates attention and is effortful, effortful mental activity that also feels like that. It's problem solving. And I find that funny. So mistakes occur if we allow System 1 to process a cognitive decision, while we should have really passed this decision over to our slow System 2. Yeah? So if we impulsively take a decision, this can be very wrong because it is not well informed and integrated with all the evidence and background information. And often it makes sense to stop your own impulse, to act quickly, but just step back and take a little bit more time, integrate more evidence, and then take the decision. So now I jump a little bit. I wanted to give you this background information because it is relevant, but now I jump to the Human Connectome project. Why do I do this? Because they have done all these cognitive tests with a large number of individuals and in a very nice and standardized manner. So this is a testing battery, and this is the lead of this Human Connectome project behavioral test setting. You see here a couple of examples. So this is, for example, the card sorting test that tests the ability for cognitive flexibility. So exactly this fast reacting to changing tasks. So, for example, first the task is to map shape to shape, so the rabbit to the rabbit, and then the task changes, map color to color, and then you have to map the orange rabbit to the orange ship or boat. And this is very easy. You do not need to solve a problem, but you need to be cognitively flexible to override the task from before and then manage to do the other task. This is cognitive flexibility. And the Human Connectome project, they have collected these phenotype information plus demographic information, psychiatric tests, and so forth. So a lot of phenotyping for each individual participant. They have done this in 1,100 subjects, and for these they also acquired structural and functional MRI, very long resting state fMRI sessions, so the entire MRI procedure lasted more than 4 hours, I think. So very high quality scans, all modalities that are required to build nice personalized brain network models, and also almost 270 and then 100 subjects with MEG. And they shared them all. So they have annotated them, they have organized them very well. They do not comply to GDPR, to just mention this, but they make the data available easily. And this is now more than 15 petabytes of data. And interestingly, more than 10,000 people used the data already. Researchers applied and signed the sharing agreement to get access to the data. and more than 800 publications came out of this nice data set. I think this is really a nice blueprint also for e-brains, how to make data useful and what actually a measure is of usefulness, namely the many people who are interested who get access to the data and to then publish papers that hopefully lead also to clinical applications eventually. So we used the data and we looked into the data. The goal was to build brain network models, but of course we also looked in the behavioral data and we found something very interesting there. So we were interested in this ability of deep thinking, reasoning, and problem solving, and therefore we looked at the progressive matrices test that I have shown you, results. PMAT, and this is the abbreviation that you find now here always. This is basically the measure, the score for fluid intelligence. And here we also have the score for the G factor that was inferred from all the other cognitive tests, that also the measure for intelligence, so they're related. But what we found, what was interesting was that this is the relationship for the first, the easy task, the relationship between the G factor, the intelligence factor, and the reaction time. And here you see that when the people were smarter, they reacted faster. And this is probably what everyone would expect, because reacting fast is beneficial, and therefore it's a sign of higher intelligence. But if we look at the most difficult task, maybe the one I showed you, I don't know whether it was number 24, but there, for all the correct answers provided, it was found that those who had higher intelligence scores, a higher G factor of intelligence, they had longer reaction times. So they took more time to solve the problem. They did not react promptly and impulsively, but they took all the time they had. There was no time limitation, and thought about the problem. But also the ones that reacted faster with lower intelligence, they had the correct answer. Yeah? But the ones who were smarter, more intelligence, they took longer to solve the problem. And this is also visible here. So here you see the 24 questions, and basically this plot and this plot, they are similar. One is individual subject results, and this is a group analysis. Doesn't matter here. But what you see, the first 8 easy questions, so the difficulty increased with each task. There we have the negative correlation. So more intelligent people, they are faster in reacting to easy problems. But then when the task got more difficult, the correlation was inverse. So the more intelligent people, according to the G factor and the fluid intelligence test result, they took more time, longer reaction time. So now we had also all the functional activity data, and we thought, and others had done that before, what is the relation now between, for example, G factor intelligence and the functional connectivity pattern. There are publications out. There is a slight, not very strong relation, which we also found, but it's not significant in our analysis. So the functional connectivity, the degree of synchronicity in the brain that is measured with fMRI, how much are the regions correlated, is not really strongly related to the general intelligence. And also not strongly or significantly related to the fluid intelligence, and also not significantly related to the processing speed test or the card sorting test, that are tests for cognitive flexibility. But when we look at the reaction times, this is the reaction times PMAT of the fluid intelligence test, here reaction time in seconds, then we see a very strong correlation with the functional connectivity. So in other words, if there's a lot of synchronicity in the brain, then the people take longer to give a response, and the responses are better. They are the more intelligent people. So there's a correlation between degree of synchronicity and the time people take to respond to a difficult problem. And this is just illustrated also here. It's the same message. So now we thought we can now build these brain network models and see what is behind it. So why does it take longer if there's higher synchronicity to react, and does it really has an effect on the outcome, on the performance, also in our models? So the first step was to just construct these brain network models and to faithfully really try to simulate the functional connectivity matrices very precisely of the individual subjects. And we had 650 data sets that had all the data that we need to do this, the behavioral data, the structural, the diffusion tensor imaging data without any artifacts and problems, and also the functional data. So we were able to use 650 data sets from the Human Connectome project, and we built 650 individualized brain network models. So now, if you have done this before, usually with a traditional approach of having mean field models that are connected via long-range excitation, you can simulate functional connectivity, but it doesn't have really a very high correlation with the functional connectivity of an individual person. So the correlation coefficients are normally not very high. So what does it mean? It means that it's not reproduced very well. The model does not really describe precisely the functional activity state or connectivity state of the brain. So we've now added some connections in our brain network model. And this is a novel aspect. This has not been done before. So what you see here, each column is representing one node in the brain, yeah? And each node consists of an excitatory and inhibitory population, and they are coupled through this long-range excitatory activity, which is provided by the structural connectome. So the excitation from one node hits the distant excitatory populations of other nodes, other brain regions. But what we added now is the input of each excitatory population to the other region's inhibitory populations. This is called feed-forward inhibition, and this is what exists in the real brain, but was not included in these models in the past. So we have now long-range excitation and long-range feed-forward inhibition in the model. And interestingly, with this, we have now a ratio of long-range excitation, long-range feed-forward inhibition that we can tune in our models. And this is illustrated here. So here you see the thick lines show a strong long-range excitation between these nodes. And the thin lines here, I don't even see my pointer now. I think the battery is empty. Oh, yeah. This shows the feed-forward inhibition, which is very, I think I take the other pointer. Which is very weak here. So here we would have a high excitation inhibition ratio in this setting. And we can change this excitation inhibition ratio by going down with the long-range excitation value and going up with the feed-forward inhibition value. So we can tune the AI ratio. And if we do this, now I'm getting confused with the controls, we see that tuning the EI ratio, you see here how we change it systematically, changes the resulting functional connectivity. So this is an easy approach to tune your functional connectivity. Yeah? If we start with a low EI ratio, we have an anti-correlation, then we go up, then we get no correlation here, and we go further up with the EI ratio, we get a strong positive correlation of the signals. So, and this is basically the degree of synchronicity or anti-correlation and correlation versus no correlation. So with the tuning now the EI ratio, it is possible to tune between all the region pairs the functional connectivity. And this is done here. You see this. We have the simulated functional connectivity and the empirical functional connectivity. We tune the excitation inhibition ratio. And here you see how the fitting improves. And we can very faithfully, for each individual subject, reproduce the empirical functional connectivity. And it is a very high degree of correlation. So if you look with the naked eye, they look identical. if you look according to the statistics, you see that if you take the traditional approach, this is very typical. You get these degrees of correlation between simulated and empirical functional connectivity. If you do the EI tuning with the long-range inhibition added to your model, you get a very high correspondence between the simulated and the empirical functional connectivity. So we can faithfully reproduce the functional connectivity of an individual subject. So now we have a model that reproduces the functional connectivity, the degree of synchronicity and the patterns, the spatial patterns of synchronicity in the brain. And we can look whether components of the model, state variables, have a relation already to the performance of the individual subjects. And we did this at the stage. And what we found was that here you see the time it took in the PMAT test and the fluid intelligence test to come to an answer. And we see a very strong correlation between the time it took to come to an answer and the input correlations that drive the nodes in our brains. Yeah? And you see an anti-correlated relationship between the time it took to come to an answer in the fluid intelligence test and the input amplitude. In other words, the higher the input amplitude in our model, that is a result of the functional connectivity tuning, the higher, the shorter was the time it took to come to a response. But the more correlated the input currents were that go to a population, the longer it took to come to a response. And this is just a group analysis, again, where we did different types of clustering. It has the same message. I do not explain that in detail. But we found relationships between the state variables, the input amplitudes, and the input correlations of the model, and the performance of the individuals. Yeah? And now we looked at the relationship of these simulated features, the input amplitude again, and the input correlation with relation to the functional connectivity, and we find a very tight relationship between functional connectivity and these model features. So now what we actually found was that when we tune the excitation inhibition balance, the long range excitation versus feed-forward long range inhibition, we not only, okay, I didn't loop this, unfortunately. Let me see whether I can start it here again. So you see that you do not only tune the functional connectivity, but also the input correlation that goes in each individual brain region of the synaptic activity and the input amplitude. So there's a maximum and then it falls off again. So now, another switch. Now I come to a circuit level model. So what I told you were brain network models that work with mean field population models, but now I switched to a circuit model that has been developed initially by Wang, sorry, and further developed by Murray and published in 2017. So the circuit model is able to mimic neuronal activity underlying cognitive performance, decision-making, and working memory. And it consists of 2 nodes in the parietal cortex and 2 nodes in the prefrontal cortex. And as you can see, they have recurrent excitation, and the recurrent excitation actually is the foundation for persistent activity or high attractor states that can be reached by these populations to mimic working memory. So if a population is stimulated and then it reaches, because of the recurrent self-excitation, a high activity persistent level, this means this neuronal activity level is a working memory trait. Yeah, it is, represents a working memory trait. On the other hand, there's also winner-take-it-all competition between these populations because they are mutually inhibiting, as you can see here. And the winner-take-it-all competition is decisive for simulating decision-making. So both populations receive sensory evidence, and it can be low contrast or high contrast, and this means how different is evidence that goes on population A and population B. And depending on which of the two populations reaches the high attractor state, decision goes to population A or population B. And what you see here, this is also important, are actually the time courses of the neuronal activity that are being simulated. I think this is helpful to see this. So here for the working memory scenario, we have one stimulus that implants a working memory in our brain, and this leads to high activity in population A, so a high attractor state. Then comes a distracting stimulus, as you can see here. And in this scenario, where the model has a certain weight in the recurrent excitation here, you see that the distractor stimulus can actually end the high attractor state of population A and establish its own high attractor state, and then the working memory trace has been overwritten by the second distracting stimulus. In this case here, where we have another weight in the self-excitation, we see that the distracting stimulus is not sufficient to delete the initial working memory trace. So we can vary the robustness of the working memory depending on the weights within the structural connectivity between these populations. Here's another example where we also vary the strength of self-excitation, and you see that for the decision-making scenario, it has impact on the speed with which the high attractor state can be reached by one of the populations. So you see here the slower, and here the population goes in a higher attractor state very quickly. Murray has explored the impact of different contrasts. So basically, if it is an easy task or a difficult task, the contrast of the evidence that goes in the one population and the other population changes. So here, for example, for the easy task, the contrast is 51%, and for the hard task, it is only 6.4%, if I can read the numbers here. And also this has an impact on the dynamics. So for easy problems, the population reacts quickly, as you can see here, and for more difficult problems, you see there's a slower ramping up of activity in the population. So now, if we take the circuit model and we look what is the impact of the different input correlations and the different input amplitude that we have found in our individualized brain network models, we see, as you can see here, that the performance, unfortunately, this is so small that not even I can see it. The correct decisions that are coded here by color, and the decision time, colored in the lower panels by color, depend on the input amplitude and the input correlation. So what does it mean? It means that either we can react fast, but then we make many mistakes, or we can react precisely, but then we are reacting slower. And at this stage, I want to summarize what we have found so far, what I have told you before. So in the empirical data, we show that slower but better solutions or decisions are linked to higher functional connectivity. Then if we simulate the higher functional connectivity in our models, this is associated with decreased input amplitudes and increased input correlations. And now if we explore the effect of decreased amplitudes and increased correlations on the circuit model, we see that there is actually an effect on the performance and on the time it takes to come to a decision. So now, since we have dynamical systems theory, we can also use the tools of dynamical systems theory and look how does the differential performance, depending on the degree of correlation and input amplitude, occur in our system. And this is described here. So what you see here is a phase portrait. So you see the activity of population B and population A. And you see the null clines, you see the separatrix, and you see 2 attractor fixed points and a saddle node point. And here you see now we move through the diagram and increase, or in this case decrease, the amplitude of activity that goes in the parietal cortex. And this reconfigures the phase space, as you can see here. And the reconfiguration of the phase space, as you can also see, changes the flow fields, and this means that the trajectory through the system changes. It has different speeds, and the time that passes until a state variable either moves to one basin of attraction or the other basin of attraction changes. So now we can look at the 3 scenarios. The high amplitude input currents linked to a low functional connectivity, so low correlation in the brain, low synchronicity. And our model is related to high amplitudes, then the default amplitude, and the low amplitude that is related to the high synchronicity, the high functional connectivity in our model. And I'm really sorry that it is so difficult to see, but what you see here are the time courses of the correct decisions for each scenario, and here the time courses of the populations that lead to the incorrect decisions. And we did 1,000 simulations, and here you see then overlaid all the responses that led to correct decisions and to incorrect decisions. And if you look closely, you will see that for the high amplitude scenario, the decision is ramping up much faster, much earlier than for the low amplitude scenario. So for the low amplitude scenario, it takes more time and it leaves more time for evidence accumulation until a decision is taken. And this is due to the phase space reconfiguration that you can see here. Okay, so this was for the amplitude, but we also saw that the degree of correlation, of input correlation changes with the functional connectivity, with the degree of synchronicity in our brain. And the correlation, the noise correlation, can be expressed in a phase space. Only as glyphs. So therefore this looks maybe a little bit unfamiliar for you, but if we don't have a fully deterministic system, but a noise-driven system, then also the field flow gradients that usually are lines, they have a distribution, and the distribution is now illustrated via these glyphs. They basically show the flow, the direction of the flow in each of the dimensions. So if we have more round glyphs, this means that the flow goes in all directions. And if we have more stretched glyphs, this means that there's more directionality in the flow. So, and now we can see if we change systematically the degree of correlation that drives our system, independence of the functional connectivity. So here you see we move this dot. We see that there's a reconfiguration of the stretchedness of these glyphs. And this means that they move from being more isotropic to more anisotropic. So that they obtain some degree of directionality. What is the impact now on our simulations? This is shown here. Same scenario. We have no correlation, medium correlation, and high correlation. And in the upper plot, we see the correct decisions. In the lower plot, we see the incorrect decisions. We have 1,000 simulations. And we can see that for the high degree of correlation in the system that is linked to higher functional connectivity, the system takes more time to come to a decision, to end up in an attractor state. And this gives more time to accumulate evidence, and the decision performance is better. Okay, and this is just a static visualization in case the previous video was too fast. So here you see very clearly that the shape of the glyphs is changing independence of the degree of input correlation. So here we have high correlations, here we have low correlations, and here you see the isotropic shape that leads, as you can see on the next slide, I think, that leads the trajectories of activity prematurely towards the two attractor states, and this is to premature jumping to a decision. While for the high correlation input, we see the stretch shapes of these glyphs, and this means that the system, the activity in population B and A stays in a balanced state over a longer time, and with this there's more time for evidence accumulation, and in this high correlation scenario all the responses were correct. So now what about the working memory? Working memory is also important and needs to be activated to keep the intermediate solutions or answers in memory to come up with a big solution for the problem. How does this react or is influenced by the degree of input currents? This is seen here. So the blue areas in this diagram indicate 100% robust working memory. The yellow areas indicate that the working memory trace is being disrupted by a distracting stimulus. And what we see very quickly with the naked eye is that for low input currents we have more blue and less yellow, and for high input currents we have more yellow and less blue, which means that depending on the input currents, the working memory trace is more or less robust. So now we have looked into all these details, and now we can bring it all together. We can use a circuit model that is capable of decision making and working memory in the full brain network model that we have tuned according to the functional connectivity of the individual subjects, that reproduces the functional connectivity of the subjects, and with this has varying input correlations and input amplitudes. So how would now the circuit model function or perform in the individualized brain network models of our 650 individuals? This is the answer. Here it is written: the brain models of smarter subjects made more correct decisions and took more time for decision making. And here you can see the statistics. So what you see here is the performance in the fluid intelligence test. And here you see the decision-making performance of our model. There's a very high correlation. And here you see the time it took to come to a decision in our model versus the performance in the fluid intelligence test of the actual subjects or persons. And this is just another visualization of this relationship. So if we have brain network models of individual persons and we plug in these decision-making working memory circuits, then they perform differently and they predict the performance of the individuals from which the models have been built. And I have shown you in the analysis of the phase space what the precise mechanism is of this differential performance. And with this I come to an end. I hope it was not too complex, and I'm very interested in your feedback and your comments and questions. I would like to acknowledge the team in Berlin, and especially the person with whom I did this particular study that I was presenting, Michael Schirner from my team in Berlin. And I thank everyone for your attention.